Imagine a world where the antibiotics we rely on no longer work. This isn't science fiction, it's a looming reality. Every year, millions of people die from drug-resistant bacteria, and the clock is ticking to find solutions. Enter artificial intelligence, AI, a tool that could revolutionize how we discover new antibiotics. But here's the catch. While AI offers incredible promise, it's not a magic wand. Let's dive into why this technology is both a beacon of hope and a reminder of the hard work still ahead. The global health crisis caused by antimicrobial resistance, AMR, is staggering. Experts predict that AMR could claim 39 million lives over the next 25 years if we don't act. Yet, despite decades of research, we haven't discovered a truly novel class of antibiotics since the 1980s. Why? Traditional methods of antibiotic discovery, like sifting through soil samples for microbe-killing compounds, are slow, labor-intensive, and increasingly ineffective. And while high-throughput screening has helped speed things up, it's extensive and often misses promising candidates. That's where AI comes in, offering a fresh approach to an old problem. In a lab in Philadelphia, robots, the size of microwaves, are hard at work assembling molecules based on instructions generated just days earlier. These aren't ordinary molecules. Some are inspired by the biological blueprints of extinct species like woolly mammoths and Neanderthals. Others are entirely new creations dreamed up by algorithms. Scientists are testing these molecules against bacteria that has grown resistant to our current arsenal of drugs. The goal? To find antibiotics capable of tackling the toughest bugs. At the heart of this effort is machine learning, ML, a subset of AI that thrives on data. ML models can analyze vast amounts of information to identify patterns humans might miss. For example, researchers feed these models data about thousands of chemical compounds known to kill bacteria, or not, and let the algorithms do the heavy lifting. Once trained, the models can sift through billions of potential compounds to suggest one's worth investigating further. As one scientist put it, ML models are suggestion generation boxes. They provide leads that scientists can then test in real-world experiments. But what makes this process truly groundbreaking is its ability to look beyond conventional sources of antibiotics. Instead of digging through dirt, researchers are mining genomic and proteomic data from across the tree of life. One lab, led by Cesar de la Fuente at the University of Pennsylvania, has pioneered this approach. His team uses ML to comb through ancient genomes and proteomes, uncovering peptides with antimicrobial potential. In one project, they identified peptides from Neanderthals and Denisovans that showed strong antibacterial activity when synthesized in the lab. Another study mined the proteomes of extinct animals like woolly mammoths and giant sloths, yielding peptides effective against infections in mice. This concept of molecular de-extinction, resurrecting molecules from long-gone organisms, is fascinating. Not only does it shed light on evolution, but it also opens the door to discovering molecules that may have protected ancient life forms and could now help us combat modern threats like AMR. Evolution encodes immense biological intelligence, De La Fuente explains. By tapping into this intelligence, we gain access to a treasure trove of possibilities. And yet, sometimes the best antibiotics may not exist anywhere, not in nature, not even in history. Consider this. The number of theoretically possible chemical compounds far exceeds the grains of sand on Earth. Most of these compounds are purely hypothetical, existing only as digital constructs. To address this challenge, researchers are turning to generative AI, which designs entirely new molecules from scratch. Unlike traditional ML models that simply analyze existing data, generative models create novel compounds tailored to specific needs. For instance, Jonathan Stokes' lab at McMaster University has developed a generative model that doesn't piece together atoms randomly. Instead, it works with predefined molecular building blocks, ensuring the resulting compounds are chemically feasible and easy to synthesize. This innovation dramatically expands the search space for new antibiotics while keeping practicality in mind. In fact, the model has already produced compounds that show antibacterial activity against pathogens like Acinetobacter baumannii. However, designing molecules is only half the battle. Turning these discoveries into usable antibiotics requires significant resources and funding. Despite AI's ability to accelerate the initial stages of drug discovery, many hurdles remain. Clinical trials are notoriously difficult, costly, and prone to failure. Additionally, antibiotics are not lucrative products for pharmaceutical companies, making investment less appealing. As both Stokes and De La Fuente emphasize, solving the antibiotic crisis will require substantial support from governments and philanthropic organizations. So, is AI the silver bullet we've been waiting for? Not exactly. While it holds immense potential, it's ultimately just another tool in our arsenal, a highly advanced pipette, as Stokes humorously puts it. Its success depends on the quality of the data it's trained on and the collaboration between scientists, policymakers, and industry leaders. And this is the part most people miss. AI alone won't defeat AMR. It takes a village to turn laboratory breakthroughs into life-saving treatments. What do you think? Can AI live up to the hype, or are we placing too much faith in technology? Share your thoughts in the comments below. After all, the future of antibiotic discovery isn't just about algorithms. It's about us.